Hi, this is the second lesson on distribution of organisms. We're looking at sampling this time, but before we get into it, pause the video and have a quick review of what you've learnt. Okay, how did you get on? Here are the answers. Again, set the bar high, 80% would be good. If you're getting less than that, have a look at the flashcards, ask questions, look back at your notes, because these are the basics that you need to know. So what are we going to learn about today? Well, we're going to learn about how we know which organisms there are in a particular area. Look at methods that we use to calculate the numbers of organisms in a particular area. And this can help us to understand, when we know the number of organisms in a particular area, that can help us to understand some of the relationships that are taking place within the community. Now that's important, particularly in conservation projects. You're looking to conserve a particular species that might be endangered or a particular habitat. It's important to understand what the interactions are between the different organisms. So we're going to look at 4 key areas. We're going to look at sampling plants, then we'll look at sampling animals, then we're going to have a look at identifying the different organisms, and then we're just going to pull the whole topic together. So pause the video, have a look at the key learning points, keep your eyes out for these points as we go through the video, and we'll review them when we get to the end of the video. So can you see a problem? If I'm asking you to go into that field and count the number of daisies in that field, is there an issue? If I ask you to go and find or count how many earthworms there are in the Broxburn area, can you see an issue? Well, there's a few issues. The first one is it'd be pretty much impossible to do. Imagine going into that field and trying to count accurately how many daisies there are there. Impossible. Count the number of earthworms in Broxburn? Impossible. Plus, you've got a life as well. I mean, how long would that take you to do? So we've got better things to do with our time. So instead, scientists use a process called sampling. This is basically where they look at a small section of a population, and then they use that information to draw conclusions about the bigger population. An example of this, if you think of, if you walk down the high street, you might see somebody with a clipboard coming towards you, or your parents, asking for their opinion. And they're sampling the population. They're asking people's opinions on a particular product, usually. And then they draw conclusions. They don't speak to absolutely everybody in the country, do they? But they'll speak to a sample and then draw conclusions from that sample. So in a similar way, scientists do something similar. They sample and are then able to draw conclusions on the whole population. So, too quick. First of all, sampling plants. So you'll have come across this high-tech piece of equipment before, a quadrat. A quadrat is basically a big square with 25 small squares within it. The key thing to remember is when you drop a quadrat onto the ground, you are not counting the number of plants. Okay? You are counting the number of squares the plant is in, not the number of plants. So, have a look at this quadrat and answer that question. Is there a greater abundance of daisies or dandelions? Okay, so what have you got? Well, if you count the plants, there's obviously more dandelions than there are daisies. But remember, when we're using this method, you do not count the plants. You count the squares the plants are in. So 1, 2, 3 would be the abundance of dandelions. 1, 2, 3, 4 would be the abundance of daisies. So please keep that in mind. You count the squares, not the plants. So if I was to go into a field, there's my field, and in that field there are dandelions and there are daisies, and I drop my quadrat down at the bottom right hand corner of the field, and I count the abundance of the plants within that quadrat, and then I draw a conclusion about the plants that are found in that field. Now, can you see a problem with me drawing a conclusion about the plants found in that field from the results I've got from that one quadrat? And how could you overcome that problem? I'll give you a subtle clue. Go on, shout it out. Repeat means? Good. Reliable means? Good. So repeat means reliable. Reliable means you repeat it. So there's my field. I've repeated it. Okay, instead of dropping one quadrat, which wouldn't give me a really good indication of the overall population, I'm dropping 5 quadrats. Okay, so can you see an issue with that, where I've dropped the 5 quadrats? Well, again, dropping the 5 quadrats in that particular area doesn't give me a good representation of the overall field, does it, of what's found within that field? So again, how could we overcome that problem? Well, spread out the quadrats, drop them more randomly, rather than just dropping them all in the same area. Okay, so generally speaking, when you're using quadrats, you will drop them randomly, and you will repeat it to make sure that the results are reliable. If you go back to the example of, you know, somebody stopping you in the street to ask your opinion on something, they try to catch as many people as they can, don't they? They wouldn't just speak to one person and then, you know, base their conclusion on what one person thinks. That wouldn't be sensible. So their sample that they take, yes, they don't speak to everybody, but they try to get as big a sample as they possibly can, because that means that their results are more likely to be reliable. So it's similar when it comes to science and carrying out these quadrats, sampling using a quadrat, that yes, we don't count everything, but the more we can repeat it, the more reliable our results are likely to be. So there are 2 main ways of using quadrats. We've talked about one where you randomly distribute them throughout an area. And that's usually used when you're comparing 2 areas. But sometimes you can use a quadrat in a systematic way. And this is called a transect, where you basically take a quadrat and you just move it along a line. So you can see in the picture here, he's at quadrat number 2 there, but he's sampled here at 1, and he's making his way along this line, 2, and then 3. You just flip the quadrant over and then count the abundance, 4, 5, 6, and that would carry on throughout the habitat. And that's usually carried out when you're looking to see the effect of one particular environmental factor on the abundance of a particular plant. So, for example, you might be looking at the effect of moisture, or light, or pH on a particular plant. Okay, so that's sampling of plants using a quadrat. So we're now going to have a look at how we can sample invertebrates. There's a definition of what an invertebrate is at the bottom of the slide. So what equipment do we use when sampling invertebrates? Well, you'll recognize this perhaps from 1st or 2nd year as a pitfall trap. So you need to be able to know how to set up a pitfall trap. There are the instructions there of how you do it. So I'm not going to go through them all, but pause the video if you need to. What I want you to do, though, is have a think about these questions. Have a think about why the pitfall trap is set up the way it is. So pause the video, have a look at the questions, and then we'll run through the answers. Okay, so why is it important for the pitfall trap to have a roof? Well, if you have a cup full of insects in the ground and no roof, any birds flying overhead will just think it's a McDonald's fly-through, shoot down fast food, and when you go to check your pitfall trap, there's absolutely nothing there. So it's just to cover up and hide the insects that you collect to stop them being eaten. Why should you check the pitfall trap regularly? Well, because insects can escape quite easily. You might end up going back to the pitfall trap if you leave it too long and you just have one big fat beetle because it's eaten everything else that was in the pitfall trap. So again, you wouldn't be getting accurate results or reliable results. Why would you put food in the bottom of the pitfall trap? Possibly to entice organisms into the trap. Why would some scientists put tiny holes in the bottom of the pitfall trap? Well, that's to allow any rainwater just to leak away, to drain away. Some scientists are evil. They put alcohol at the bottom of the pitfall trap or antifreeze, and that's basically to kill the insects as they fall into the pitfall trap, and that stops them from escaping, and it means your results are more likely to be realistic. So, what do you think about Bob? Bob set up a pitfall trap in a field, and he finds no insects in the pitfall trap. So Bob's conclusion, based on his sampling, is that there are no insects in the field. Now, is that likely? Is that a realistic expectation that there are no insects at all in a field? Well, no, obviously not. So what does Bob need to do to make his results more reliable? Oh, talk about banging on about a point. He needs to repeat it. He needs more than one pitfall trap. He needs to do it again and again and again, and he's more likely to get reliable results. So we've talked about sampling plants. We've talked about how we sample invertebrates, but we have another issue. Imagine you found this in your pitfall trap. What would you call that thing? Well, you might call it a beetle, but there are more than 4,100 beetles in the UK. What happens if you found this in your pitfall trap? What would you call that? Well, you might call it a bee, but there are more than 20,000 different types of bee in the world. You find this in your pitfall trap. What are you going to call that? Again, you might call it a slug, but there are 40 different types of slug in the United Kingdom. So you find this plant. You throw your quadrat and there's this plant. What are you going to call that plant? What is that plant? Do you know what that plant is called? Probably not. It's a plantain. Did you know that? Well, how do we identify these things? If there are 40 different types of slug, how do we know which slug it is that's in our pitfall trap? If we don't know all the names of the plants, how do we know which plants there are in our quadrat? So that's an issue that scientists and biologists face. How do you identify the things that you're sampling? You can't just Google it. So instead, we use something called a paired statement key. So here's an example. So if you imagine that you found this invertebrate in your pitfall trap, to use the paired statement key, the paired statements, so at each stage, stage 1, stage 2, stage 3, and stage 4, you're given a choice. And you have to decide which one you're going to choose and then follow the instruction on the right-hand side. So if we take a look at this as an example, So the first paired statement, the choice is, does it have 6 legs or does it have more than 6 legs? If you look at the picture, it's obviously got more than 6 legs. So we follow the instruction, go to 2. So we go to 2. Now follow the instructions there. Does it have 8 legs or does it have more than 8 legs? Oh, it's got more than 8 legs, so follow the instruction, go to 4. So we're skipping 3. Does it have 1 pair of legs per body segment, or does it have 2 pairs of legs per body segment? So there are the segments, and it's got 1 pair of legs per body segment. So the answer is chilopoda. So that we've identified that insect, that invertebrate, by using the paired statement key. Wouldn't have a clue what it is without using the key. So have a go. Pause the video and see if you can identify A, B, C, D, E, and F using the picture on the right-hand side. Okay, so how did you get on? Here are the answers. Mark and check that you've got that right. Just check this one here. Make sure you got that right. If you want more practice, here's another example, identifying the fish. So use the paired statement key on the left-hand side to identify the fish on the right-hand side. Pause the video now, and I'll show you the answers in a minute. Okay, so there are the answers. Again, if you're not sure, if you're struggling with this, make sure you ask questions. Okay, last little section that we need to cover today is basically the definition of what an indicator species is. Quite a tricky little bit because it's a tiny section of the course, easy to forget. So try and find a way of remembering what an indicator species is. So as you're sampling, if you come across particular species in an area, it can give you an indication of how polluted that area is. So for example, I've given you 2 organisms. One of these organisms lives in very, very clean water. And one of these organisms lives in very, very dirty water. So one of these organisms can only live where there are very high levels of oxygen in the water, whereas the other one can live when the oxygen levels are quite low and it's very polluted. And I reckon you can work out which one is which. So if I was sampling a river and I found a rat-tailed maggot in the river, that would indicate, see what I did there? That would indicate that the river is polluted because rat-tailed maggots are able to survive in very polluted water. If I find a mayfly nymph in the river, that would indicate that the river is very, very clean because they're only able to survive where the oxygen levels are really high. Okay, so by their presence or by their absence, they indicate the levels of pollution in a particular area. So let's pull everything together from this topic. Pause the video. Have a look at the data, have a look at the information, and have a think about the questions at the bottom, and then we'll run through the answers. Okay, so how have you got on? So what is the abiotic factor being measured? Well, if you take a look, abiotic meaning non-living. So the non-living factor that you're measuring here is the moisture. Which quadrat? So quadrant 1, quadrant 2, quadrant 3, quadrant 4, or quadrant 5, which quadrant is likely to be closest to the river? Not 100% sure, but probably quadrant 1, because the moisture reading is highest there. Not necessarily, but probably quadrant 1. Which plant is in most need of the abiotic factor? So which plant, so come over to this part of the table, is it plantain, clover, or grass? Well, grass can cope when the moisture is high and when the moisture is low. So that is not in need. The plantain, however, you can see that the abundance of plantain is extremely high when the moisture is extremely high. And the plantain is extremely low when the moisture is extremely low. So I would say the answer for that would be plantain. Which plant is least affected by the abiotic factor being measured? Well, I would say that was grass, because as you can see, whether the moisture is high or whether the moisture is low, the abundance of grass is extremely high. And can you spot any incorrect readings and explain? Did you spot it? That reading there is incorrect. It has to be wrong. Why? Because the highest abundance you can have using a quadrat is 25, because you're counting the squares and not the number of plants in an area. So have a look at the key learning points. Just check that you can explain these points to somebody else. That's how you really know you understand it. So can you explain to somebody how to sample plants in an area, how to use a quadrat? Can you explain how to set up a pitfall trap in order to sample invertebrates? Can you use and make a paired statement key to identify things, organisms? Can you remember what the definition is of an indicator species? And that last slide that we've just looked at, really, can you see how we can link these things together, abiotic factors and sampling, and we can look at reasons why plants and animals live where they do.